Welcome, curious minds, to The Deep Dive. Today we're going to challenge, well, maybe even upend your fundamental understanding of inheritance.
Yeah, it's a big one.
We usually think of heredity as, a fixed blueprint, simple passing down of genes, maybe a few random mutations here and there.
It's a standard Mendelian view, basically.
Exactly. But what if that blueprint isn't quite as fixed as we imagine? What if, say, the experiences of a parent, or maybe even a grandparent, could leave a real lasting imprint on their offspring?
Shaping traits, influencing health.
All without changing a single letter of their actual DNA code.
It sounds almost like science fiction, doesn't it? But it's very real science. It falls under the umbrella of epigenetics.
Epigenetics, right.
And the term itself isn't actually new. It was coined way back in 1942 by Conrad Waddington.
Wow, 1942.
Yeah, initially he used it in developmental biology, trying to describe how cells could develop specific features and create diversity, even though the exact molecular nuts and bolts weren't known then.
Okay, so the meaning has shifted a bit.
It's evolved, definitely. Today, when we say epigenetics, we're talking about the specific molecular mechanisms that allow different gene states, different levels of activity to be perpetuated using the same underlying DNA sequence.
Gotcha. So our mission to this deep dive is to really plunge into this idea of epigenetic inheritance. This frankly, mind-bending concept that things like environmental factors, life experiences,
diet, stress, toxins,
can actually cause physical changes, phenotypes that get passed down, sometimes across several generations. It really broadens how we think about heredity.
Absolutely. We're going to unpack this whole emerging research area. We'll look at some really surprising evidence, both in mammals and, interestingly, in plants, too.
And we'll dig into the molecular tools, the actual biological machinery that makes this possible.
And then think about the impact, right? What does this mean for health, for disease, maybe even evolution? And something quite relevant today, how we grow our food.
Consider this your shortcut to getting really well informed about a concept that is fundamentally reshaping biology. We'll have surprising facts, maybe a little bit of humor to keep you hooked. And for this, we've drawn on some really cutting-edge sources, scientific reviews like Epigenetic Inheritance, Impact for Biology and Society, another one called Epigenetics, possible applications in climate-smart crop breeding.
Plus, articles looking specifically at non-Mendelian heredity, how plants manage this. Yeah, a good mix.
So get ready. This might just expand your whole understanding of life.
And it's not just abstract science, is it? This is about understanding how your own environment, maybe even your ancestors' environment, might be subtly influencing your biology right now.
For anyone curious about the future of food, which should be all of us, really, it reveals some genuinely groundbreaking possibilities for creating more resilient crops in our, well, rapidly changing climate.
Definitely food for thought, pardon the pun.
Okay, let's unpack this then. We're talking about this epigenetic layer beyond the basic DNA sequence. What exactly is this layer? How does it manage to, remember things like experiences?
Right. So think of your genetic code, your DNA sequence, as this enormous, incredibly detailed instruction manual for building and operating you.
Okay, the master blueprint.
Exactly. And that sequence, the letters A, T, C, G, is pretty much fixed. What epigenetics refers to is this additional layer of information, modifications that sit on the DNA or on the proteins the DNA is wrapped around.
So not changing the words in the manual, but adding notes in the margins.
Highlighting.
Sticky notes.
That's a perfect analogy. Sticky notes, highlights, annotations. Telling your cells which parts of the manual to read, maybe how loudly or how often to read them, or which parts to ignore completely.
And these notes aren't permanent. They can change based on life.
That's the key. They are dynamic. They can be influenced by environmental factors, diet, stress, all sorts of things. And crucially, sometimes these notes can be copied and passed down to the next generation.
Mind-bending. Okay, so what's the molecular toolkit? How are these notes actually written and read?
Let's start with the most studied mechanism. DNA methylation. This involves adding a tiny chemical tag, specifically a methyl group that's CH3, to certain letters in the DNA sequence, usually to cytosine bases, particularly when they're next to a guanine base, the so-called CPG sites.
Like adding a little off switch or maybe a dimmer switch onto a specific instruction?
Yeah, dimmer switch is often a good way to think about it. It generally tends to repress gene activity, make it harder for that gene to be read.
Okay, so who are the molecular writers, the enzymes putting these methyl tags on?
We have a whole crew of enzymes called DNA methyltransferases, or DNMTs. Some, like MT1 in plants, are really important for maintaining existing methylation patterns when DNA replicates. They basically ensure the sticky notes get copied onto the new DNA strand.
So the memory persists through cell division.
Exactly. Others, like DRM2 in plants, are involved in establishing de novo methylation, putting brand new tags on places that weren't tagged before. And interestingly, DRM2 is often guided to its target spots by small RNA molecules.
Small RNAs, like little GPS signals telling the enzyme where to write.
Precisely. It's part of a pathway called RNA-directed DNA methylation, or RDDM, a very elegant system. And there are other methyltransferases too, like CMT3 and CMT2, which handle methylation in slightly different DNA contexts, adding layers of complexity.
Okay, if there are writers, there must be erasers, right? Ways to take these methyl marks off.
Absolutely. The system needs to be dynamic. In plants, we have specific DNA glycosylases enzymes with names like ROS1, DME, DML2, DML3. They recognize a methylated cytosine, snip the bond holding the methyl group, and then initiate a repair process to restore the original cytosine.
Do they peel off the sticky note?
Essentially, yes. In mammals, a different family of enzymes called TET enzymes handles this, oxidizing the methyl group as a first step towards removal. While the exact plant equivalents aren't fully pinned down, we see the chemical byproducts suggesting a similar process exists. And interestingly, putting human enzymes into plants does cause demethylation.
Fascinating. And the main effect of methylation is turning genes down.
Primarily, yes. It often acts to repress transcription. It can do this by physically blocking activator proteins from binding to the DNA, or by recruiting repressor proteins that actively shut the gene down.
Like cluttering up the page so you can't read it easily?
Kind of. Sometimes methylation within the gene's coding region can lead to messed up transcripts or silence potentially problematic transcription start sites, but it's not always repression. Less commonly, methylation can actually activate gene transcription, though exactly how that works is still a bit murky.
And you mentioned other types of DNA methylation, not just on cytosine.
Right. While cytosine methylation, 5-MC is the classic one, there's also N6-adenine methylation, 6-MA. It occurs in plants, too, and seems to be associated with genes that are actively expressed. What's really interesting is that 6-MA appears to be highly responsive to environmental stimuli, adding yet another layer to this epigenetic code.
Okay, so we've got tags directly on the DNA. What about the proteins associated with DNA? You mentioned histones earlier.
Yes, histone modifications. So remember, our DNA manual is incredibly long. To fit inside the cell nucleus, it's wrapped around proteins called histones, like thread around spools. A segment of DNA wrapped around a core of 8 histones is called a nucleosome.
The basic packing unit.
Exactly. And these histone proteins have tails that stick out, and these tails can be chemically modified in dozens of different ways. These modifications profoundly affect how tightly the DNA is wound around the histones.
So this controls how accessible the DNA is. Like if it's packed super tight, the machinery can't get in to read the genes.
Precisely. If the chromatin, that's the DNA histone complex, is tightly packed, genes are generally silenced. If it's loosely packed, genes are accessible and can be activated.
And what kinds of modifications are we talking about?
Lots. Acetylation is a common one, adding an acetyl group. This usually loosens the chromatin and is associated with gene activation. Think of it like unspooling the thread a bit.
Makes sense.
Then there's methylation again, but this time on the histone tails. Adding methyl groups to specific amino acids on the histones, like lysine 4 or 36 on histone H3, is often linked to gene expression. But methylation on other sites, like lysine 9 or 27 on histone H3, is usually associated with gene repression and the formation of that tightly packed inactive chromatin we call heterochromatin.
Wow, so methylation can mean go or stop, depending on where it is on the histone.
Exactly. Context is everything. And what's really key is that DNA methylation and histone modifications don't work in isolation. They often talk to each other, they cooperate. Regions with low DNA methylation often have histone marks that promote transcription, and vice versa. It's an intricate interplay, a sort of histone code layered on top of the genetic code.
Okay, DNA tags, histone modification.
Yeah.
What else is in this epigenetic toolkit? You mentioned non-coding RNAs earlier.
Yes, non-coding RNAs, ncRNAs, are turning out to be major players. These are RNA molecules that don't code for proteins themselves, but have crucial regulatory roles. One important class is small interfering RNAs, or siRNAs.
The GPS guides for DNA methylation.
Some of them, yes, particularly in plants. They're tiny RNA molecules, maybe 21 or 24 nucleotides long. They can guide the methylation machinery to specific DNA sequences, effectively silencing gene expression. They are absolutely critical for keeping potentially damaging elements in check, like jumping genes or transposons.
Like genomic security guards.
A good way to think of it. Then we have things like transfer RNase-derived small RNAs, or tsRNAs. These fragments of transfer RNAs have been found to change in sperm in response to things like diet. And intriguingly, some studies suggest these changes can sometimes be reversed by altering diet again or through exercise.
Showing that real-time responsiveness.
Definitely. And microRNAs or miRNAs are another class. They primarily regulate gene expression after the gene has been transcribed into mRNA, often by degrading the mRNA or blocking its translation into protein. Some studies suggest they can be influenced by things like reactive oxygen species, or ROS, produced by mitochondria. Many miRNAs found in sperm actually originate from mitochondria themselves.
Connecting metabolism and energy production to epigenetic regulation. Fascinating. What about longer ncRNAs?
Right. We also have long non-coding RNAs, or lncRNAs. These are transcripts longer than 200 nucleotides. They're incredibly diverse. Some can act as scaffolds, bringing proteins and other molecules together in the nucleus to regulate gene expression. Others can interact directly with DNA or chromatin.
More layers of control.
And even more recently, circular RNAs, or circRNAs, have emerged. These are formed by a weird back-splicing process where the ends of an RNA molecule join together, making a stable loop.
Circular. Why?
Well, that circular structure makes them very resistant to degradation compared to linear RNAs. Their exact functions are still being worked out, but some can act like sponges, soaking up miRNAs, thereby influencing the expression of genes targeted by those miRNAs. There's even evidence suggesting DNA methylation might play a role in how these circRNAs are made.
It just keeps getting more complex.
And one final layer, RNA methylation. Just like DNA, RNA molecules themselves can be chemically modified. The most common is N6-methyldenosine, or M6A, on messenger RNA. These marks can affect mRNA stability, how long the message lasts, as well as splicing, processing, where the mRNA goes in the cell, and even how efficiently it's translated into protein.
So the notes aren't just on the DNA manual, but on the temporary copies too.
Exactly. It's this incredible multi-layered system. DNA tags, histone modifications, a whole zoo of non-coding RNAs, even modifications on the RNA messages themselves. All interacting, all dynamic.
It really paints a picture of biological flexibility far beyond just the static DNA sequence we used to focus on.
Absolutely. It's a constant adjustment, a fine-tuning of gene expression in response to countless internal and external signals. That's the essence of epigenetics.
Okay, here's where it gets, for me anyway, really interesting. What happens to one generation? Diet, toxins, stress, maybe even behaviors ripple through to the next. Sometimes for multiple generations, all without changing the A's, T's, C's, and G's.
Yeah.
How does that biological memory get transmitted?
It's the million-dollar question, isn't it? And you're right, this phenomenon, transgenerational epigenetic inheritance, really pushes beyond classical Mendelian genetics. For a long time, the central dogma was basically genetics is everything for heredity. There was this strong belief, almost a dogma itself, that the germline, the sperm and egg cells, was somehow pristine, shielded from environmental influences in a way that could be passed down.
The Weismann barrier idea, right? That somatic changes don't affect the germline.
Exactly. That view really dominated for decades. But over the last, say, 20 years, that perspective has shifted dramatically. Thanks to better animal models, incredible advances in sequencing technology, our ability to actually measure epigenetic marks in sperm and eggs, we now have compelling evidence that the environment can leave heritable marks on the germline.
So the barrier isn't absolute. Or maybe there are ways around it.
It seems less absolute than previously thought, or perhaps there are specific windows or mechanisms that allow information to cross. We're seeing this play out in real-world examples, both in humans and animal models.
Okay, let's hear them. What are some concrete examples in mammals where these echoes from the past show up?
Let's start with a really striking human study on paternal smoking and asthma risk. This research, led by Cecilie Svanes in Norway, used a huge data set, 35,000 people across 4 generations.
Wow, that's massive.
It is. And the key finding was stunning. The risk of non-allergic asthma in children was significantly higher if their father had started smoking before the age of 15.
Before 15. Not just smoking before conception, but specifically starting that early.
Exactly. Starting smoking before puberty had a much stronger association with offspring asthma risk than starting later, even if they'd whipped before conception. And this early paternal smoking was also linked to lower lung function in the kids and, importantly, detectable changes in DNA methylation in the offspring's blood cells.
So something about that prepubertal window makes the developing male germline particularly sensitive to the effects of smoking, leaving a mark that affects the next generation's respiratory health.
That's precisely what the evidence suggests. It highlights these critical windows of susceptibility. Prepuberty is emerging as a really important and previously quite understudied period where environmental exposures can potentially induce lasting changes in the maturing sperm stem cells. And it wasn't just smoking.
Oh, what else?
The same study found that if the father became overweight starting in prepuberty, that also increased the likelihood of non-allergic asthma in his children, even independent of the child's own weight.
That's incredible. So for you listening, it really hammers home that your health isn't just about your own choices and your direct genes, but potentially about your parents' exposures, especially during these key developmental times.
It adds a whole new dimension to thinking about health inheritance.
Absolutely.
Yeah.
Okay, what about something as basic as diet? We hear so much about diet affecting our own health. But can dietary patterns really leave a multi-generational legacy?
The evidence from animal models is certainly pointing that way. Take a study by Adam Wilkins looking at a low-protein diet in male mice. Feeding males this diet actually led to a loss of DNA methylation in their sperm.
So erasing some of those epigenetic notes.
Yes, and it also altered the levels of the enzymes that write those notes, the DNMTs, and changed the expression of metabolic genes right there in the testes. But it wasn't just the sperm.
What else?
The seminal fluid produced by these males was also different, and this is fascinating. When they performed IVF using both the altered sperm and the altered seminal fluid, the resulting offspring showed all the same phenotypic changes seen when mating occurred naturally. But using just one or the other only produced partial effects.
Wow, so it suggests an additive effect. Both the sperm's epigenetic state and the environment it's delivered in contribute to the outcome in the offspring.
Exactly. It highlights that the transmission might be more complex than just what's inside the sperm head. Now, flip the coin to a paternal, high-fat diet, HFD. Nora Kotaj's lab studied this in mice. Males on HFD gained weight, got glucose intolerant, as you expect.
Standard stuff.
Right. But their immediate offspring, the F1 generation, were interesting. Under normal conditions, they were fine. But when they were challenged with a high-fat diet, they actually gained less weight than control offspring.
A protective effect. Inherited metabolic resilience.
It looked like it initially, almost like the father's unhealthy diet pre-programmed the offspring to handle fat better. But then they looked at the grand offspring, the F2 generation. They showed higher insulin levels. So the effect wasn't simply protective. It shifted across generations, revealing a more complex, potentially detrimental consequence later on.
That's intricate. Did they find any molecular clues, like those RNA changes we talked about?
Yes, they found specific changes in those transfer RNA-derived small RNAs, tsRNAs, in the sperm of the HFD fathers. And here's another twist. These tsRNA changes could be partly reversed if the fathers went back to a normal diet or exercise.
Okay, showing some reversibility. That's good.
But, and this is a big but, when the HFD fathers were treated with metformin, a common drug for type 2 diabetes, the tsRNA changes in their sperm actually got worse.
Worse. With a diabetes drug.
Yes. It raises really important questions about how clinical treatments might have unexpected, potentially negative impacts on the epigenome of germ cells and thus on future generations. We need to consider these transgenerational effects when evaluating drugs.
Definitely a cautionary tale. Okay, moving from diet to environmental toxins. Can exposure to harmful chemicals also leave these long-lasting echoes?
Absolutely. And this is a major area of research. Michael Skinner has been a real pioneer here, working on this for over 20 years. He coined the term generational toxicity.
Generational toxicity, what does that entail?
His key observation, particularly with endocrine-disrupting chemicals, is that the effects are often relatively minor in the individuals directly exposed, maybe during embryonic development, the F0 or F1 generation.
So the first generation exposed might seem okay?
Relatively speaking, yes. But then pathologies become more pronounced in later generations, like the F1 and especially the F3 generation. And since the F3 generation was never directly exposed to the toxin,
it strongly implies the effect was transmitted through the germline sperm or egg across the F1 and F2 generations.
Exactly. It suggests a germline-dependent transgenerational effect. The proposed mechanism is that the initial toxicant exposure induces stable epigenetic changes like altered DNA methylation patterns in the germ cells of the exposed individual.
Right.
These altered epigenetic marks are then passed on, modifying gene regulation during the development of the subsequent generations. This leads to altered gene expression patterns and epigenetic states in both the body cells, somatic, and germ cells of those descendants, ultimately increasing their susceptibility to disease later in life.
Can you give a specific example?
Sure. Emily Rissman's work with bisphenol A, BPA, a common chemical found in plastics, is a good one. Ancestral exposure of mice to BPA led to altered social recognition behavior and changes in gene transcription in the brains of their F3 grand offspring.
3 generations later from the initial exposure.
Yes. And it also caused physical changes in the brain, like an increase in the density of dendritic spines on hippocampal neurons. And this effect was dose-dependent. Crucially, they saw these transgenerational effects in 2 different strains of mice, suggesting it wasn't just some quirk of a specific genetic background.
Which strengthens the epigenetic argument.
Definitely. And the implication here is huge for how we assess chemical safety. Standard toxicology often only looks at the directly exposed generation. Skinner and others argue we need to take a much longer view, looking across multiple generations, and incorporate this systems epigenetics perspective. Epigenetic marks could even become biomarkers, early warnings of disease risk passed down from ancestral exposures.
It really changes the timescale of environmental health impacts. But is it all negative? Can beneficial things, positive experiences or interventions also be passed down epigenetically? That feels important.
It's a critical question, and thankfully the answer seems to be yes sometimes. Research by Berman's Iskandar looked at folic acid supplementation in adult rats and mice. Folic acid is a B vitamin known to be important for development.
Right.
They found something surprising. Supplementing the parents' diet with folic acid actually enhanced the ability of neurons in their offspring to regenerate after injury. Specifically, their axons grew back better.
Okay, a beneficial effect passed to the next generation. That's cool.
But here's the truly remarkable part. This enhanced regeneration capacity wasn't just seen in the immediate progeny. It persisted in subsequent generations of animals that were never exposed to extra folic acid themselves.
How long did it persist?
Up to the 6th generation.
Just 6 generations? Seriously?
Seriously. And the beneficial effects seem to accumulate additively across those generations, especially in genetically diverse outbred animals.
That is absolutely incredible. A positive trait accumulating over 6 generations passed down epigenetically. Do they know how?
They correlated it with changes in DNA methylation and gene expression related to axonal growth, particularly evident in the F3 generation. Another interesting finding was that the maximum benefit in the offspring was seen when both parents had received the folic acid supplement, suggesting both the male and female germlines contributed additively.
Both parents matter.
Yes, and they could even reverse this inherited trait. Treating the later generation animals with the drugs that inhibit histone deacetylase or demethylating agents brought the regeneration capacity back down to baseline levels.
Showing it really was epigenetic and potentially malleable.
Exactly. So the big takeaway for you, the listener, is that this epigenetic inheritance isn't just a story of doom and gloom, of passing down risks from toxins or poor diet. Positive interventions, nutritional support. These might also leave a beneficial, lasting epigenetic legacy across generations.
That's a much more hopeful note. Okay, let's switch gears now to the plant kingdom. We've touched on it, but do plants show similar kinds of transgenerational memory, passing on experiences with stress, for instance?
Oh, absolutely. In many ways, epigenetic inheritance is even more widely studied and perhaps better understood in plants. It makes sense, right? Plants are rooted in place. They can't run away from stress like drought or heat. So having mechanisms to remember past stresses and prime their offspring for similar conditions would be highly advantageous.
Kind of inherited resilience baked in.
Exactly. A great example comes from studies on stress memory in Arabidopsis, that little model weed scientists love. Kay Brzezinka and colleagues looked at heat stress priming.
So giving the plant a little taste of heat makes it tougher against future heat waves.
Precisely. And they identified a specific gene they called forgetter1, or FGT1. The protein made by this gene binds to other genes that get switched on during heat stress. By binding there, it seems to modify the chromatin, the DNA packaging, keeping those heat response genes sort of poised, accessible, and ready to be activated quickly if another heat stress comes along.
So FGT1 helps the plant not forget the previous stress, maintaining that preparedness.
That's the idea. It contributes to the stress-induced chromatin memory, and this memory can sometimes be passed on.
Okay, what about a major stress like drought, especially over multiple generations?
There's compelling evidence there, too. Work by Zhang and colleagues looked at multi-generational drought adaptation in rice. They found that drought stress induced specific changes in DNA methylation. They called these epimutations.
Epimutations.
And these altered methylation patterns were actually maintained in successive generations of rice plants that were also exposed to drought. This strongly suggests that these heritable, drought-inducible epimarks are part of how rice populations adapt to dry conditions over time.
Passing down the drought survival guide.
In a molecular sense, yes. And another example in barley from Keser Donja's team. They found that drought stress led to increased DNA methylation in the promoter region, the on-off switch of a specific gene called cytokinin oxidase 2.1, HVCKX 2.1.
Okay, what did that do?
Seeds collected from these drought-stressed barley plants showed faster shoot emergence when they were planted. So, an epigenetic change induced by drought in the parent plant was directly linked to a potentially beneficial trait getting established faster in the offspring.
That's a direct link from epigenetic mark to useful trait. Huge implications for breeding, surely.
Immense. These findings really underscore the vital role of epigenetic modifications in how plants respond to stresses like drought and how they adapt long-term. It's the foundation for this idea of climate-smart crop breeding, trying to leverage these epigenetic mechanisms.
How does the transmission work in plants? Is it different from mammals, given they have things like pollen?
It is quite different in some key aspects. Unlike mammals, where the germline is set aside very early in development, plant germ cells, the precursors to pollen and egg cells, arise much later, often from mature plant tissues, post-embryonically.
So those tissues have experienced the environment directly.
Right. And while there is some epigenetic reprogramming, some wiping the slate clean during plant reproduction, it seems to be less complete than in mammals. More epigenetic marks, more memory seems to get carried over from one generation to the next.
Allowing that environmental experience to be passed on more readily.
Potentially, yes. If we look at pollen development, the male germline gives rise to 2 sperm cells and a vegetative nucleus. The sperm DNA is supercondensed, mostly inactive, while the vegetative nucleus has looser chromatin and active genes. Small RNAs, particularly 21 nucleotide siRNAs in mature pollen, and 24 nucleotide siRNAs that appear later in the seed embryo, are crucial guides. They direct the inheritance and reestablishment of methylation patterns, especially for silencing those transposons, those jumping genes. There seems to be an ancient mechanism involving waves of demethylation and remethylation during fertilization to ensure transposons are recognized and kept quiet in the next generation.
Wow. So plants have this incredible capacity to remember environmental stress and pass that memory on.
It's truly remarkable, and it has profound implications, not just for understanding how plants survive in the wild, but potentially for how we can purposefully breed crops to be more resilient in the face of climate change. It's about tapping into their natural epigenetic adaptability.
Okay, so thinking about inheritance more broadly now, what does all this mean? It really feels like there are more pieces to this puzzle than just the DNA sequence inside the sperm and egg. You mentioned the seminal fluid earlier.
Yeah.
How else can experience or environmental information get transmitted across generations?
That's exactly where a lot of cutting-edge research is focused. We're realizing that the picture is much richer and more complex than just the germ cells in isolation. For instance, we're looking closely at the somatic cells within the gonads and accessory reproductive structures.
So not the sperm or egg themselves, but the cells around them, like support cells.
Precisely. Cells like the Sertoli cells and Leydig cells in the testes, which support sperm development, epididymal fat cells, newer sperm mature, nursing cells in the ovary, even the composition of the seminal fluid itself. Studies are showing that these components can also be significantly modified by environmental exposures, things like diet or toxicants.
And sometimes these somatic cells are affected even more strongly than the germ cells.
Yes, that's been observed. It suggests that these different cell types might have different vulnerabilities or perhaps different protective mechanisms, maybe related to membrane receptors, internal signaling pathways, or the effectiveness of barriers like the blood-testis barrier or the blood follicle barrier.
So the big question is, can changes in these surrounding somatic cells, or in things like seminal fluid, actually influence the epigenetic state of the sperm or egg? Or maybe even contribute directly to the phenotype of the offspring, bypassing the germ cell's own epigenome?
That's the frontier. The evidence is building that these somatic components can indeed communicate with and potentially modify the germ cells or contribute signals that influence early development after fertilization. It adds layers of complexity to how environmental information gets captured and transmitted.
Okay, what other unexpected players are emerging in this expanded view of inheritance?
Well, mitochondria in sperm are a fascinating and quite unexpected example.
Mitochondria. But I thought sperm mitochondria were destroyed after fertilization, so they don't contribute their DNA to the embryo.
That's the traditional view, yes.
Oh.
Paternal mitochondrial DNA is generally eliminated. However, recent research, particularly using fruit flies, Drosophila, by Anita Ust and her group, suggests sperm mitochondria might still play a role in epigenetic inheritance.
Wow. If they don't pass on their genes.
They found that dietary challenges in the male flies led to changes in the small RNA composition of their sperm, and these changes were dependent on the sperm mitochondria. Remember, mitochondria are the cell's power plants, producing ATP, but they also produce reactive oxygen species, ROS, as a byproduct.
Right, ROS can be signaling molecules.
Exactly. The idea is that mitochondria, influenced by the father's diet, can alter the sperm's internal environment. They might change mRNA expression through ROS signaling. They could also modify the populations of those tsRNAs we mentioned earlier, possibly through differential activity of mitochondrial genes, because it turns out many tsRNAs found in sperm actually originate from mitochondrial tRNAs.
So the father's metabolic state, reflected in his sperm mitochondria, could alter the RNA cargo delivered to the egg, even if the mitochondria themselves don't persist.
That seems to be the hypothesis. It's a way for the father's metabolic experience to leave an imprint. And interestingly, they found dose-dependent effects. With sugar concentration, for example, a mid-range dose induced molecular changes in sperm, while very low or very high doses didn't. We see similar nonlinear dose responses with endocrine disruptors, too.
Highlighting the need to test a range of exposures, not just high doses.
Absolutely. So even if eliminated, sperm mitochondria affected by the environment could mediate cellular effects that contribute to epigenetic inheritance, acting independently of the nuclear DNA.
Okay, what about signals traveling through the body? Could something in the bloodstream influence the germline?
Yes, and that brings us to extracellular vesicles, or EVs. These are tiny membrane-bound packages released by cells that can travel through the bloodstream carrying proteins, lipids, and crucially, RNA molecules.
Like little messages sent between tissues.
Exactly. Groundbreaking work from Isabel Mensu's lab showed this connection. They took blood serum from male mice that had experienced early life stress and injected it into control non-stressed males.
And what happened?
The injected males showed changes in the RNA signatures within their sperm, and remarkably, their subsequent offspring exhibited some of the same stress-related behavioral and metabolic symptoms as offspring from directly stressed fathers.
Whoa. So the stress signal was transferable through the blood serum.
It appeared so. Further experiments using purified EVs from the blood of stressed males confirm this. Injecting these EVs intravenously into control males altered the transcriptome, the pattern of active genes in their sperm.
Just from the EVs?
Just from the EVs. And again, the progeny conceived from these males showed altered gene expression at the very early two-cell embryo stage and developed metabolic symptoms typical of stress exposure when they grew up.
That's incredible. It suggests EVs circulating in the blood can directly signal the male germline, carrying information about the father's experiences, like stress, and contributing to the transmission of those traits to offspring.
It really opens up a systemic view, where signals from potentially anywhere in the body could theoretically reach the germ cells via EVs and influence the next generation.
Mind-blowing. Okay, one more piece. The epididymis. This is the tube where sperm mature after leaving the testes.
Yeah.
Does that environment play a role?
It certainly seems to. Research by Brett Nixon looked at the effects of acrylamide exposure in mice. Acrylamide is a chemical found in some cooked foods and industrial processes.
Okay.
They found that acrylamide exposure changed the protein composition and the cargo of small non-coding RNAs, essentially RNAs, not only in the sperm themselves, but also within the epithelial cells lining the epididymis, particularly in the caput or head region where early maturation occurs.
So the maturation environment itself was altered by the exposure.
Yes, and this seemed to have consequences. The glucocorticoid receptor, GR, involved in stress responses, was altered in the epididymal tissue, and this correlated with changes in specific mRNAs known to regulate GR. When sperm that had matured in this altered environment were used for IVF, the resulting embryos showed abnormal gene expression patterns and often failed to develop properly.
Suggesting that the sensi-RNA cargo picked up by sperm during their passage through the epididymis is crucial for early development, and that this cargo can be compromised by paternal exposure.
Precisely. It adds another layer. The post-testicular environment matters too. So when you put it all together, influences from somatic support cells, mitochondrial signals, circulating EVs, the epididymal environment, it raises this fundamental question.
Yeah.
If factors outside the germ cells themselves can significantly modify the information passed on to the next generation, how does that force us to redefine heredity? It's moving way beyond just the DNA sequence in the gametes to a much more integrated whole organism view of inheritance. We're really just starting to map out this incredibly complex landscape.
It's fascinating. All right, let's dive back into the plant world now. We've seen how epigenetics helps plants remember stress, but how does it orchestrate their normal development and contribute to the huge diversity of traits we see, and even shape their evolution over millions of years?
It's absolutely central to plant life. DNA methylation, for example, plays incredibly dynamic roles right from the earliest stages of development. One of the most striking examples is genomic imprinting.
We touched on this, where a gene's activity depends on whether it came from the mother or the father plant,
Right.
controlled by methylation.
Exactly. It's a parent of origin effect. In flowering plants, angiosperms, fertilization is actually a double event. One sperm fuses with the egg cell to form the diploid zygote, which develops into the embryo, but a second sperm fuses with another cell, the central cell, which already has two nuclei, forming a triploid tissue called the endosperm.
Triploid, 3 sets of chromosomes.
Yes, typically 2 from the mother, from the central cell, and 1 from the father, from the second sperm. And the endosperm's main job is to nourish the developing embryo. Genomic imprinting is critical for regulating the development of this endosperm.
How so?
There's this idea called the parental conflict theory. It basically suggests that paternally inherited genes tend to promote endosperm growth, maximizing resource allocation to their offspring, while maternally inherited genes tend to restrict endosperm growth, perhaps to conserve resources for the mother plant to produce more seeds later.
A genetic tug-of-war over resources for the seed.
Pretty much. And DNA methylation is the referee, or maybe the tool they use. Before fertilization, the central cell, maternal precursor, undergoes significant demethylation removal of methyl tags at specific genes.
So the maternal copies get wiped clean, ready to be active.
Exactly. As a result, after fertilization, in the triploid endosperm, the maternal alleles of these imprinted genes are generally less methylated and therefore more highly expressed, while the paternal alleles remain methylated and silenced.
Can you give examples of these imprinted genes?
Sure. Genes like flowering Wageningen, FW, Medea, and fertilization-independent seed2, FS2, are classic examples. They get demethylated in the maternal central cell, so they're expressed from the maternal copies in the endosperm after fertilization. These genes are crucial regulators of seed development. MEA and FIS2, for instance, are involved in chromatin remodeling and gene silencing.
And FWA affects flowering time, too, you mentioned.
Yes, its imprinting in the endosperm is linked to seed development, but proper silencing of FWA in other tissues is critical for a normal flowering time. If it gets abnormally demethylated and expressed elsewhere, the plant flowers very late. A key enzyme here is the demethylase DME, which is active in the central cell, maternal side, and also in the vegetative nucleus of the pollen, paternal side in some cases, specifically removing methylation marks from these imprinted genes, and also, interestingly, from some mobile elements or transposons.
Why demethylate transposons there? Isn't silencing them usually the goal?
It seems counterintuitive, but one hypothesis is that temporarily activating these mobile elements in the cells surrounding the gametes allows small RNA molecules to be produced from them. These small RNAs can then be delivered to the actual egg and sperm cells, where they act as guides to reinforce the silencing methylation on those same transposons in the gametes, ensuring they stay quiet in the next generation.
Wow, like a controlled burn to generate the blueprint for future fireproofing.
That's a great analogy, and this whole system of genomic imprinting seems particularly prevalent in plants that have the most common type of embryo sac development, the polygonum type, found in over 70% of flowering plants.
So it's a widespread, fundamental epigenetic control mechanism for the very first stages of plant life, balancing parental contributions. Amazing. What about traits we can actually see, like flower color, scent?
Epigenetics, especially DNA methylation, has a direct hand in phenotypic expression, the visible characteristics of plants. Floral pigmentation is a beautiful example. Think of Oncidium orchids. There's a popular yellow variety, Gower Ramsay, and a white one, White Jade. The difference comes down to the methylation state of a single gene, OGCCD1. This gene codes for an enzyme that breaks down carotenoids, the yellow pigments. In the yellow orchid, the promoter of OGCCD1 is heavily methylated, so the gene is silenced, the enzyme isn't made, and the yellow pigments accumulate.
Makes sense. And in the white one?
In the white orchid, the OGCCD1 promoter is unmethylated. The gene is active, the enzyme is produced, it degrades the carotenoids, and voila, white flowers.
So simple yet elegant.
It is. We see similar things controlling red streaks in other orchids via methylation of anthocyanin pathway genes, or the way petals on some crabapple trees change from red to pale pink as the promoter of a key pigment gene, MHMYB10, becomes progressively more methylated during flower development, shutting down pigment production.
It's like the plant is painting itself using epigenetic controls. What about smell?
Even floral scent. In the Japanese plum, Prunus mum, researchers found that genes responsible for producing key volatile scent compounds show dynamic changes in DNA methylation levels as the flower develops. This directly influences the blend and intensity of the fragrance produced at different stages.
Incredible. And presumably less visible but vital processes too?
Yes, while maybe less visually dramatic, there's growing evidence that epigenetic marks also influence fundamental processes like photosynthesis, tuning the efficiency of how plants capture light energy.
So epigenetics is deeply woven into the fabric of plant development and traits. How does it play into the bigger picture of plant evolution?
It plays an ancient and fundamental role. Remember those transposons, the jumping genes? The mechanism for silencing them using DNA methylation is found across the entire plant kingdom, from ancient mosses right up to modern flowering plants. This tells us it's a deeply conserved, essential defense system for maintaining genome stability that evolved very early on.
A core function from the beginning.
Absolutely. And the regulation of ordinary gene expression by DNA methylation is also a central feature across all land plants. But then, as plants evolved, particularly with the rise of flowering plants, angiosperms, epigenetics took on new, more complex roles.
Like innovations built on that ancient foundation.
Exactly. That RNA-directed DNA methylation RDDM pathway, especially the version involving a specific enzyme called RNA polymerase V, seems to be more elaborate in angiosperms. It's been hypothesized that this pathway helped plants deal with the consequences of whole genome duplication events, which happened quite often in plant evolution, perhaps by helping to silence redundant gene copies and streamline the genome.
Helping manage genomic complexity.
Yes. Also, the maintenance of methylation in non-CG contexts by the enzyme CMT3 appears to be an angiosperm-specific feature, adding another regulatory layer. And as we discussed, genomic imprinting is widely considered a major evolutionary novelty, pretty much unique to flowering plants, enabling that sophisticated parental control over seed development.
So the epigenetic toolkit itself evolved and became more sophisticated over time.
It seems so. And the overall level of DNA methylation varies quite a bit between different plant species. It often correlates with how much repetitive DNA and how many mobile elements a plant has in its genome species, with larger, more complex genomes tend to have higher overall methylation levels, presumably to keep all that non-coding DNA in check. This suggests DNA methylation itself is an evolutionary trait linked to genome size and complexity.
It's truly incredible to step back and see how these subtle molecular modifications have played such a profound role in shaping the entire diversity, development, and survival strategies of plants over vast stretches of evolutionary time, from a flower's color to its ability to cope with a changing world.
It really underscores how dynamic and adaptive life is, using these layers of regulation beyond just the raw DNA sequence. It's a constant dialogue between the genome and the environment, written in epigenetic ink.
Okay, this is all fascinating, understanding the mechanisms and the evolutionary sweep. But let's bring it back to the present and future. If we understand these intricate epigenetic mechanisms, can we actually use this knowledge? Can we harness epigenetics, especially for something as critical as feeding a growing population on a changing planet?
That's the huge potential and the focus of a lot of current research. The idea of climate-smart crop breeding is exactly that. Leveraging what we're learning about epigenetics to deliberately enhance crop adaptation and resilience.
So instead of just traditional breeding based on crossing plants with good genes, we'd also be selecting for plants with beneficial epigenetic marks.
Precisely. The goal is to improve tolerance to stresses like drought, heat, salinity, cold, even heavy metals in the soil, all major challenges amplified by climate change. We want to create climate-smart crops that can maintain yields even under these tougher conditions.
How do you even go about selecting for epigenetic traits? They seem less stable than DNA sequence changes.
That's a key challenge, which we'll get to. But one approach is to exploit the natural or induced epigenetic variation that is reasonably stable and heritable. A powerful tool being developed is the use of epigenetic recombinant inbred lines, or EPIRL.
Epirales, what are those?
Okay, imagine you start with two plants that are genetically almost identical, but they differ significantly in their epigenetic profiles, perhaps because one was derived from a plant with altered DNA methylation. You cross them and then breed the offspring for many generations, similar to creating standard RILs for genetic mapping. But because the underlying DNA sequence is largely the same, any consistent trait differences you see in the resulting epigenetic population are much more likely to be due to the inherited epigenetic differences rather than genetic variation.
So it helps isolate the effect of the epigenetics from the genetics.
Exactly. It reduces the confounding factor of DNA sequence differences, allowing breeders to more effectively identify, study, and potentially select for beneficial epillelies these heritable epigenetic states.
Clever. Are there specific crops where this is already showing promise?
Yes, definitely. In maize, researchers are finding that certain histone modifications like H3K4ME3 and H3K27E3 might act as memory marks for genes involved in responding to stress. They're combining data on gene expression, transcriptomics, with chromatin data like histone modifications and DNA accessibility to get a really detailed picture of how maize responds to drought. This is revealing important roles for non-coding RNAs and specific epigenetic patterns in drought tolerance.
And rice. You mentioned the multi-generational drought adaptation.
Right. That study in rice showed that drought-induced epimutations in DNA methylation were maintained across generations, correlating with improved drought adaptability. It's a clear case of heritable epigenetic memory providing a benefit.
And barley.
In barley, that finding about drought-increasing methylation at the HVCKX 2.1 promoter, leading to faster shoot emergence in the offspring seeds, that's another direct link between an environmental stress, an epigenetic change, and a potentially useful agronomic trait. Our sources actually provide tables listing numerous examples across many crops.
Like wheat, soybean, tomatoes.
Peas, chickpeas, rapeseed, alfalfa, showing various epigenetic mechanisms, histone changes, methylation, demethylation, RNA accumulation, being involved in responses to a whole range of abiotic stresses. The potential is clearly there.
It sounds incredibly promising, almost like a new green revolution based on epigenetics. But what are the hurdles? It can't be straightforward.
No, definitely not. There are significant challenges in studying and applying epigenetic inheritance for crop improvement. One of the biggest is the issue of stability and heritability.
We touched on that the sticky notes aren't always permanent.
Exactly. While some stress-induced epigenetic modifications seem quite stable and get passed down reliably for several generations, many others are transient. They might revert back to the original state once the environmental stress is removed. For breeding purposes, you need traits that are reliably heritable. So a lot more research is needed to understand what makes some epiallels stable and others unstable.
Okay, stability is key. What else?
Related to that is the fact that traits heavily influenced by epigenetics often show lower heritability in the classical genetic sense. This makes them harder to select for using traditional breeding methods that rely on predictable inheritance patterns. Plus, these traits can be very sensitive to environmental fluctuations, leading to unpredictable performance in the field, which is obviously a problem for farmers.
Makes sense. More variability, less predictability.
Then there's the fundamental challenge of disentangling epigenetic effects from underlying genetic variation. The genetic sequence itself can influence where epigenetic marks are placed, and the stability or effect of an epimutation might depend on the specific genetic background of the plant line.
So nature and nurture are deeply intertwined, even at the molecular level. How do researchers try to separate them?
It requires sophisticated approaches. Multi-omics is key, simultaneously measuring the genome sequence, the epigenome like DNA methylation patterns, and the transcriptome gene expression levels, often now even at the single-cell level. This gives a much more integrated picture. And advanced gene and epigenetic editing tools like CRISPR-Cas9 systems adapted for epigenome editing are becoming crucial. They allow researchers to make very precise changes, adding or removing a specific methyl group, for example, without altering the DNA sequence, to directly test the causal effect of that epigenetic mark.
So you can experimentally prove that changing the sticky note changes the trait.
That's the goal, moving from correlation to causation. Another challenge, more on the statistical side, is something called the Beavis effect in genetic mapping.
The Beavis effect.
It is. It refers to a statistical bias, particularly when working with smaller experimental populations. When you're mapping quantitative trait loci, QTLs, regions of the genome associated with variation in a trait, you might overestimate the effect size of the QTLs you do manage to detect. It's a form of winner's curse. You only detect the apparently strongest signals, which might be inflated by random chance.
So you might think an epigenetic mark has a huge effect when it's actually more modest.
Potentially, yes. The way to combat this is by using very large populations for mapping studies and employing robust statistical methods, potentially including machine learning algorithms, to get more accurate estimates of the true effect sizes.
Okay, one last challenge mentioned was loss of heterozygosity, LOH. How does that fit in?
LOH is another non-Mendelian phenomenon. It happens when a heterozygous individual having two different alleles for a gene loses one of those alleles in some of its cells, becoming effectively homozygous for the remaining allele.
How does that impact breeding?
It can be a double-edged sword. On the one hand, LOH can reduce genetic diversity within a population, potentially decreasing adaptability or leading to inbreeding depression if harmful recessive alleles are unmasked.
That sounds bad.
It can be, but on the other hand, LOH can actually increase uniformity within a plant variety, making all the plants more similar, which is often highly desirable for commercial agriculture. It can also inadvertently speed up the breeding process by leading to rapid fixation of certain alleles in hybrid lines, getting you to a stable homozygous state faster than expected by Mendelian rules.
Like that rice example you mentioned earlier.
Exactly. The AMR rice plant caused widespread LOH in its hybrid offspring, leading to near-complete genotype fixation in just the F2 generation, much faster than normal segregation would predict. They attributed it to unusual chromosome behavior during mitosis called assortment mitosis. So LOH is another layer of genetic complexity that can interact with breeding efforts, sometimes helpfully, sometimes not.
Wow. So the path to truly harnessing epigenetics for crop improvement is complex.
It absolutely is. While the promise is immense, truly, imagine crops inherently equipped to handle the stresses of tomorrow's climate. We're still very much in the phase of moving from fascinating descriptive observations to deep mechanistic understanding and demonstrating clear cause and effect. Achieving reliable, predictable epigenetic engineering for agriculture is still a significant hurdle. But the potential payoff is so huge, it's driving incredible innovation in the field. It's early days for widespread application, but the potential is genuinely transformative.
What an incredible journey we've taken today. We've explored this amazing, almost hidden world of epigenetics, seeing how our experiences, signals from the environment, even the internal chatter within ourselves, can leave these lasting marks stretching across generations. It really does redefine inheritance.
Absolutely. From impacting our health based on maybe our father's teenage habits, to giving plants the memory to withstand drought, this hidden layer is clearly a profound and dynamic force in all of biology. It shows us that the story of life isn't just etched in the stone of DNA sequence, but constantly being written and rewritten in the margins by experience.
This deep dive really underscores that inheritance is so much more complex, so much more fluid than just the genes we get from our parents. And it makes you wonder, if the experiences of our ancestors, these intricate gene-environment interactions, can shape us and the living world around us in such profound ways, what does that mean for us? What new responsibilities does this knowledge bring?
That's the big question, isn't it? It touches on our personal health choices, the environments we create and are exposed to, the way we approach agriculture, our connection to past and future generations. It's a lot to think about.
And the field is moving so fast. As you said, there's still more questions than answers.
Definitely. We're just scratching the surface of understanding these intricate biological memories and how they work. But every new discovery offers a more nuanced, more fascinating view of life.
So the message is, stay curious.
Yeah.
Because the next breakthrough in epigenetics could truly redefine our understanding of life itself and our place within it.